Hi, in this video, we're continuing to explore immune repertoire analysis by considering gene usage. Gene usage is actually a powerful metric allowing to assess the diversity and biases of the immune response. So to start with the gene usage analysis, I'm selecting the gene usage block here and adding it to the project. So here we are selecting the data set that we're going to analyze. In case of this example data, in the clonotyping, we profiled all 7 genes. So we are selecting the one here that we're going to analyze. If you need to analyze multiple genes, then you need to add multiple gene usage blocks. So in the parameters, we have grouped by either allele or gene, meaning that we are either calculating the usage for allelic variants of genes or for genes themselves. So by default, we are using genes. We are clicking the Run button to start the analysis. In fact, gene usage analysis should be pretty fast. It's a simple calculation, and we will get result quick. So once calculated, we have our usage heat map. Let's make it larger. So this is basically the usage. On the y-axis, we have genes. If we zoom in, then we will start seeing the actual gene names. I don't want to zoom in so much, because there are many genes. We don't see them by default. So to explore the gene usage, let's go to the data mapping and start bringing our metadata into the analysis. So specifically in this project, we have a time point as a main metadata column describing, giving the context for our downstream analysis. So I'm going here, and in the metadata, we have time point. So let's use time point to actually, we can use the metadata for various purposes. So we can use time point, for example, to put an annotation layer and see samples annotated with on the x-axis annotated with our time point to see whether we have a clusterization. Actually, by default, you see this heat map has a hierarchical clustering for genes and for samples. And first of all, we see that there is a nice grouping of samples in clusters according to the time point, which is pretty obvious because here in this specific study, we have one patient, multiple time points, and in each time point, we have multiple samples. So obviously samples are grouped since they are picked at the same time point. So, we can enrich our visualization by changing, for example, the color palette. We can change the colors for the annotation layer. We can do many different things to improve our visualization and export it for publication. But let's return to the data mapping. So why we use gene usage? We use gene usage to identify whether there are some biases or patterns in our, let's say, to characterize the immune response against something, or to understand how the immune system responds to specific pathogens, specific disease, or specific events. And that's the case for this study. And how I'm going to do this, I'm going to use time point actually to not only annotate the x-axis, but also to group our samples by the time point. And now you see a very nice picture. So we have time points and samples group and time points. And on this plot, you clearly see that at the 8th day after immunization 1, we have this specific V gene really prevalent. Let's go to the data mapping and put best V gene to the tooltip. So now we see the actual best V gene. We can also navigate, copy, and we see that IGHV315 is the one that is expanded at the 8th day after vaccination, which is the result that we are actually expected here. As you remember, in the repertoire diversity, our previous guide, we identified that at the 8th day after immunization 1, we really have expansion of our immune system response. So the clonality. So we have this gene identified as the one which is involved primarily into the immune response. So let's go further and see what we have else here. So in the same way, we can bring other metadata to the heat map. We can play around with the heat map to do different things. But let's explore the controls. What we have out here, we have weighted and unweighted. So it's important. Weighted means that the gene usage is calculated using the abundance of every clonal. So here, the value of the heat map is actually there, corresponds to the total abundance of a specific gene in a specific sample. So the value is the abundance, measured in terms of the unique molecular identifiers. That's what we have for this data, or the number of reads, if it's just non-UMI data. And if we switch to the unweighted, then we actually will see the same heat map. Well, not the same heat map, the heat map where the color corresponds to the number of such genes, not to the abundance. Yeah, so it's unweighted, but just to the count of such genes in sample. And we can do the same actually. We can use our time point as a group to see whether we see the prevalence of the certain genes. And yes, we see. We can bring same way the best gene to the tooltip, and yeah, we see that the same IGHV315 is the one that is prevalent at the 8-day after immunization one. So this is a nice scientific result. Uh, the same we can observe on the G-gene usage plot. So here you see sections, V-gene usage and G-gene usage. Here we have the same for G-genes. Let's switch back to the weighted. And for the weighted, let's do the same. Let's use time point as a grouping to see whether we have such pattern in for the V-gene, for the G-gene usage. And well, clearly we see some different patterns. So specifically at the 8-day after immunization one, we have this G gene expanded, while the others are less prevalent. So this is just an example. Depending on your biology and on your project, you will be looking at different things. And finally, the VG gene usage is a combination. It's loaded. Is a combination of V and G genes. On the x-axis, we have V genes. On the y-axis, we have J genes. And here we can switch between different samples, because this combination we can see only for individual samples. And for each sample, we can see what is the pattern of the combined VG gene usage. Same way, weighted and unweighted. So that's it. This is gene usage analysis. And stay tuned for the next guys for further downstream analysis blocks.